Hello again. It's twice a pleasure to see you all in the same day. And where is Joe? Is Joe still around? Hi, Joe. It is once again my delight to speak with you all about the work that we do, and more broadly to share with you a bit about marine microbes. Now, if I were to go into marine microbes in detail, none of you would leave here tonight, and it'd be about another week before we could all head out. There's a vast amount to cover. I'm going to give you some highlights in a few minutes that I hope will help you develop a better understanding of where microbes are, how abundant they are, and the role they play in running our planet. Now, to set the stage, I wanted to start by introducing to you to a few definitions and a few figures from history. But what is a microbe or a microorganism? Put simply, it is organisms that are smaller than we can see with our naked eyes. They are distinct from macroorganisms or megaorganisms, but they're an extremely diverse group, and I'll come back to that later. When we say microbes, microbes is a catch-all. Microbes is not that different than saying living things, right? Living things applies to a lot. Microbes applies to a lot of living things that are too small for us to see. It does not imply that they are all very closely related. Some of them are more so than others, but I hope that you kind of get the sense of where I'm going at here. They're quite diverse. Now, they can live by themselves or they can live in groups. Just because they live in groups doesn't make them an organism, an animal in the way that we are an animal. But microbiology really began a long time ago, and it didn't really sort of gel until about the 1600s when Robert Hooke was the first person to describe microorganisms. He was looking at the fruiting bodies of molds through an objective here. And then, of course, perhaps more famous than that was Antony von Leeuwenhoek, who basically looked at the first bacteria using his little microscope, which, by the way, if you're interested, you can still buy a replica of today. You can look up the Leeuwenhoek microscope on any number of the auction sites and the like, and you can get your own, and they're lots of fun. But he was the first to describe bacteria. And so he put to paper his observations about these little animalcules, as he called them, or these microscopic organisms that are beyond our ability to see. It was throughout the 1600s and 1700s that scientists began to move away from this idea that life could spontaneously generate. Because they would see, for example, worms growing on rotting flesh or mold covering cheese, and the idea was maybe that life could spontaneously arise. And it was the work of Pasteur and others who really demonstrated that that wasn't the case. But I want to take us now to environmental microbiology and tell you about two folks who are incredibly important in our understanding of microbiology, in as much as they arguably launched environmental microbiology. And that's Martinus Beijerinck and Sergei Winogradsky. So I'm going to tell you briefly a bit about these folks, but Beijerinck worked actually to develop techniques to grow specific microbes in labs. Now, we take this all for granted, but in the 1800s, Beijerinck was really interested in finding a way to get particular kinds of microbes to grow while preventing other kinds of microbes to grow, and he developed what we call selective media. He was also the fellow who coined this phrase that everything is everywhere. This is a common theme in microbiology, the idea being that if there's a microbe that makes a living by, let's say, eating a particular kind of chemical, if that chemical is available in soil and the conditions are right, it'll grow there. If that chemical is available in water and the conditions are right, temperature, oxygen, whatever, it'll grow there. So the idea is that microbes are universally, or at least biospherically, to coin a silly phrase, distributed, right? That they're everywhere. And I'll come back to that in a little bit. But Sergei Winogradsky actually furthered our understanding of microbes by recognizing that some microbes could make a living from chemical energy. I briefly alluded to this earlier, but chemical energy is everywhere around us. Anytime you take a match to your gas barbecue or gas stove, you're starting a poorly controlled reaction, right? You are taking something that has a lot of electrons, in this case methane, and you are igniting it in the presence of oxygen, right? And so the electrons essentially move from one compound to the other, creating different kinds of compounds and releasing heat. And that's what, allows us to grill our veggie kebabs and the like. There are organisms in this world that have biochemical pathways that can capitalize off the energy tied up in methane and in hydrogen and in hydrogen sulfide, that nasty rotten egg smell that comes out of sewers. That's a great energy source. There's an incredible diversity of microbes that can do an incredible diversity of things. And I think this is as good a point as any in this presentation to tell you that microbes can do things that we animals simply cannot do. We animals, humans, in terms of our energy metabolism, are, quite frankly, boring. Now, I apologize to any doctors or any human physiologists out there, but really the way you make your energy, or the way you harvest your energy or harness your energy, is not that different than every other animal on Earth. Your energy metabolism is fundamentally the same as a goldfish, and a newt, and a zebra, lion, lizards, any of those creepy deep-sea fish I showed you. It's true. You all have to eat stuff, organic matter, and I don't mean whole foods, I mean organic matter, and you breathe oxygen, and you harness your energy from the oxidation of that organic matter. That's what you do. But the bacteria and the archaea, which I'll tell you a little bit more about later, are microbes that don't necessarily need to do that. Some of them can live off of these gases that are toxic to us. Many of them can live without oxygen. Some of them can live in places that we couldn't imagine. Do you know there are microbes that can grow in nuclear reactors? It's true. And we always think, well, man, how do they survive that? How do you survive that? Especially because radiation damages DNA, among many other things. But if you damage your blueprint, how do you deal with that? Well, they have a dozen copies that they maintain at all time, and they error correct, right? You take all of them and you figure out what's busted and what's not, and that's how they do it. So microbes are mind-blowingly cool in terms of their physiological abilities. But it wasn't that long ago that we didn't think microbes lived in the ocean. And in fact, it wasn't until the middle of the 20th century that a fellow named Claude Zobel and colleagues went out to the ocean and collected seawater samples from the deepest depths and found that they could grow microbes on plates of agar, right? The stuff that you get from seaweed. By giving them a little bit of food, they could get colonies to grow. And they realized also, Claude Zobel in particular, that some microbes require pressure, and called them barophiles or piezophiles, organisms that required pressure to make a living. And this was a real game changer. And so that really launched the field of marine microbiology when we began to recognize, in fact, there are plenty of microbes in the ocean. Well, who are these microbes and where do they live? Let me give you a brief introduction. Microbes live everywhere. Now, if you think about the biosphere, which I alluded to previously today, as the habitable volume of our planet, where animals live is fairly restricted. Because, for example, there's no animal on Earth that can live above temperatures of about 55 or 60 degrees Celsius, 125 Fahrenheit or so. You can't. It doesn't happen. You can deal with it for a little while. On my sillier moments, I'll reach into boiling water and grab a potato. That doesn't make me a thermophile or thermotolerant, right? But there are microbes that can live at temperatures well above the boiling point of water because they live down at the deep hydrothermal vents where the pressure keeps water from boiling at the temperatures we're used to. In other words, microbes that live above 100 degrees Celsius. There's no animal on Earth that can do that. There are microbes that live in the cracks within ice and in the deep subsurface. So essentially, where they live is everywhere. I would argue that they define the limits of our biosphere, not animals. But I'm often asked, well, how many microbes are there in the ocean? And this is a lot of fun because this has been a hot topic as of late, especially starting about 30 years ago, and then it sort of reemerged 10 years ago. About 30 years ago, or a little bit more, before PCR was really commonplace, we would count microbes by either trying to grow them on a plate or by looking at them under a microscope. Now, if you take seawater and you streak it out on a plate, that is, by streak I mean you take a sterile plate and you rub seawater on it, you give them some food, you get these dots. Now, these dots are individual bacteria that have settled and have grown into a colony of clones. And so that's great. You count the dots and you say, okay, I can get about 100 dots, which should represent about 100 cells from 1 milliliter of seawater. But then if you take that same milliliter of seawater and you put it on a microscope slide and you look at it with something that stains them, and you start to count, you actually realize you get about 1 million. This is called the Great Plate Count Paradox, and for many years it irritated people. It's like, well, how come we can only grow 100 if there's actually 1 million in there? And that's what I meant by the role of PCR in furthering our understanding of the microbial world. How many of you are familiar with PCR? See a show of hands. How many of you have not heard of it? Okay. So for those of us who geek out in the lab with our microbes and trying to figure out who's there, one of our most powerful tools is the ability to take a living group of cells, say 1 milliliter of seawater, and using some chemical processing to pull out the DNA. And we put it in solution. Now, for the most part, but not entirely, but for the most part, in order for me to know who's there, I have to take that DNA and make many, many, many copies of it. In order for me to get enough DNA that I can put it into a device called a sequencer and actually figure out the genomic sequence of that DNA. That's facilitated by a chemical reaction called the polymerase chain reaction, or PCR. It's the basis for all the DNA fingerprinting you see on your television crime shows. It's how we're able to take pieces of DNA out of one organism and put them in another, for better or for worse. But it's a very powerful tool. And I should tell you that the irony is not lost on me that PCR exists because someone found out how a microbe that lives in a hot spring duplicates its DNA. So the actual enzyme that we use, which we nicknamed TAC, for those of you who have heard of PCR, you may have heard of TAC polymerase. That name TAC doesn't mean like a thumbtack. It actually stands for Thermus aquaticus, the microorganism from which this particular gene and then protein was isolated. So it was a microbe's ability to replicate its DNA in a hot spring that allowed us to replicate the DNA of countless other microbes in the lab. And that was a mind-blowing event in our understanding of the world around us. What you see here is what we call a phylogenetic tree. This is basically the sequence of a bunch of microbes that are connected by lines, the distance of which represents how related they are to one another. That's it. It's like, think of it as a genealogy, right? If you're here on the genealogy, someone closer to you is more closely related to you. This is not that different, right? But before we were really good at PCR, we had this kind of worldview. I learned this, it's funny, I think of myself as not that old until I have a birthday. But I remember, in the 80s, sitting there in school learning this and thinking, oh, yeah, okay, there's plants and fungi and animalia, and then you got these Protista and the Monera, the so-called, bacteria and microorganisms. And we had this idea that this is how things were related to one another. And it's absolutely not the case. Because of PCR, we're able to amplify the same sort of chunks of DNA out of many, many organisms, and we see something emerge like this, which tells us that the bacteria that we know and love are their own group, as are the Archaea. These are also microbes, but they are actually distinct from the bacteria. These two are the ones that do all those amazing things I was telling you about physiologically. And then there's the Eukarya. These are the Eukaryotes. And so all of these little lines here are things like yeast and amoeba and all that. And guess where we are? All the plants and animals and fungi that you're aware of are right at the tip there. So this fundamentally changed our worldview about the diversity of microbes on Earth, and has led to many, many interesting new discoveries. For example, some of the biochemical features of these Archaea are very closely related to us. So so some people say, well, we must have somehow descended from this Archaea, or something that was Archaea-like. And this is a big hot topic. We also know that your power plants, the mitochondria in your cells, you may have heard of these, the little power plants that keep you going, they're very closely related to a kind of bacteria. So you probably started off as a symbiosis. Well, not you. Well, you too, but I'm muddling things up left and right. But back in time, Eukarya started off as a kind of symbiosis. And we, of course, are now symbiotic with microbes, which I'm going to tell you all about momentarily. But again, people say, well, all right, there's a lot of microbes. When people say there's a lot of microbes, what does that mean? Well, there are an estimated 10 to the 27th microbes in the ocean. And if you assume that each microbe is a micron in diameter and it weighs about a femtogram, which is about right, what we find is that they weigh about 10 to the 15th grams, and we weigh about 10 to the 14th grams, all of humankind. But this is what blows my mind. If you put every microbe in the ocean end on end, like a little string, they would stretch 10 to the 21st meters. Well, that's a big number, right? And I can't wrap my head around that. It's the tyranny of zeros. Sorry. It's like, it's so many zeros, I can't understand it until I calculated it in light years. It's 105,000 light years. So if I took all the microbes out of the ocean and made a string, it wouldn't just go to the moon, a mere 250,000 miles away. It would stretch across our Milky Way.
That's a long way.
Yeah. So I, for one, have given up on talking about astronomical numbers because I talk about microbial numbers now, right? These gigantic numbers, the gigantic number of microbes on Earth. I wish Carl Sagan were still around. This would be a fun fight to pick, you know? Because they are truly tremendous in number. And this is the important point. We tend to dismiss microbes because they're small, but then we don't dismiss them when they cause illness in us. Then we get all uppity about it, right? And of all the microbes on Earth, the vast diversity, there's probably millions and millions of different kinds. I'm not going to say species because that concept doesn't hold for microbes. But tens of millions of different kinds of microbes. About how many do you think cause human disease? Thousands?
No.
About 200 at best. Right? So we have this huge bias. And we look at microbes and we think germs. Right? But without most of the microbes on Earth, you wouldn't be here. And I'll explain that in a little more detail here. But when we look at the ocean, we can see that the highest density of microbes are actually at the surface. And their density decreases as we go down with depth. This is just depth. It's called a Semilog scale. Don't worry too much about it, other than you start off at about 1 million microbes in a milliliter. So milliliter is about the size of the tip of your pinky, right? That's what you get at the surface. And when you're down in the deep sea, you're down to about 1,000. So it's 3 orders of magnitude, or 1,000 times fewer microbes per milliliter of water. Nevertheless, as I mentioned, the microbes are distributed throughout our biosphere. And I'm going to take you on a quick tour of some habitats to give you a sense of where microbes are. So there are the so-called Bacterioplankton. These are microbes that are beautiful and diverse. They include bacteria and archaea and eukarya. Those include things like amoeba and algae and so on. And they're floating around in the ocean. So when I talk about 1 million cells per mil, I'm talking about bacteria and archaea, and then there's a few other tens of thousands of the algae and the like. Okay? That's what we find floating around in the water. When you get to the seafloor, we find that microbes tend to form carpet-like things called mats that drape over features on the seafloor. This is one of my favorites. This is a tiny, tiny chimney off the coast of Palos Verdes in about 600 meters of water in the shipping lane that we discovered about 2 years ago. Right, because I was on the search for hydrothermal vents off Los Angeles, and we didn't find the kind of vents we found, but we found these. And this is basically where methane was bubbling out of the ground, and the microbes have contributed to building this carbonate rock. And they basically form this thick, luscious biofilm or microbial mat on top of them. But microbes are also found, to our knowledge, in just about every creature on Earth. Now, there are some interesting organisms that don't seem to have microbes in their guts. There are some ants, for example, that seem to have microbe-free guts. We don't really know what that means. But the vast majority of organisms have microbes in their guts that play a huge role in their health. Now, you may have heard there's more microbial cells in you than there are U cells. It's probably actually not true. Sorry to bust that neat little, pseudo-fact. But it's not about the numbers. It's about the role they play. And it's really clear that they play a role in your nutritive health, right? It's also true for other animals in the world. Now, this is a funny story because this is a project that I got me hooked up with Joe, and it's been a real fun effort, and it's dragged me willingly into the world of marine mammals. For most marine biologists, you don't want to work on marine mammals, especially when you're an undergraduate or graduate, because pshaw, that's just so amateur. No, I study deep sea tube worms, or I study the real animals. And marine mammals, everybody wants to study flippers, so you get really you get really sort of snotty about it, right, or self-righteous. But marine mammals are really cool. And one of the things that that happened was a few years back, an undergraduate approached me and said, I want to look at the kinds of microbes in whale poo. And I thought, well, why not? Let's give it a go. And one thing led to another, and as soon as we started working closely with Joe, who really is the expert on the whales and their ecology, and being trained originally as an animal physiologist, we got really excited about this. Joe and I started thinking, well, what does this all mean? And we began to look at this whale's microbiome. And to make a really long and interesting story short, these whales are descended from animals that are like cows and camels, right? So they have chambered guts, but they're actually predators, right? They're not eating plants, they're eating tiny little animals. So, they're, from that point of view, they're more similar to lions and tigers, although it certainly seems like a really unfair match. But that's probably a better way of describing. So we predicted their gut would look like a carnivore's, right? And in fact, part of it does. But the other thing that we saw is they have a bunch of microbes in their gut that break down chitin, which is what makes the shells of these little crustaceans. And that is really similar to the kinds of microbes in cows that break down cellulose. They're different microorganisms, but it tells us that maybe they're using their chambered foregut in a way that we hadn't really considered. Like, what if one of these chambers is dedicated to these chitin-degrading bacteria that need an oxygen-free environment? So, without diving too deeply into the history here, the whole microbiome world has been thinking about what matters most, your history or your diet. And here's an example that shows us the relevance of both. There's a history that is responsible for that chambered gut, and the fundamental change in their diet means we see different microbes that capitalize off that gut morphology, if you will, that gut shape. But microbes also have a lot to do with whales when they're dead. Right? And Joe mentioned this as well, that whales are a heck of a major nutrient fall in the deep sea. And they host communities that are really seemingly unique to whales. Those of us who studied hydrothermal vents and related environments in the deep sea for a long time thought that whales were a stepping stone for vent animals. Like my tube worms that I love at hydrothermal vents, maybe they could grow on whales and get to the next vent in some way. And there's very little data to support that being commonplace. But what we know is there are lots of organisms that are endemic, meaning unique to whales. And that's an interesting thought. Organisms that evolved seemingly to live specifically on whale deadfalls or other deadfalls. There are also some really extraordinary examples of microbial animal relationships. I mentioned the tube worms. These tube worms are near and dear to my heart. Does anybody know what this is? It's a Riftia tube worm. Their name is Riftia pachyptala. They live at hydrothermal vents. You've probably seen pictures of these on any number of the television show, sort of documentary TV shows. But this is a red feathery plume, and their body lives inside these white tubes that they make. The tallest tube I've seen is about this tall. The worm inside it was about yay big. It has the consistency of, like a kielbasa, right? And this is in the deep sea where many things are very flabby and gelatinous because there isn't a lot of nutrition and isn't a lot of energy, and you have kind of watery tissue. Not these worms. These worms are big and robust and very active, and they live around hydrothermal vents in the eastern Pacific. They have no mouth, no gut, no anus, no digestive tract. Instead, they host somewhere between 1 billion and 1 trillion bacteria deep inside their bodies that they feed chemicals to, in this case hydrogen sulfide, and those bacteria take those chemicals and use the energy to convert carbon dioxide to sugar and feed themselves and the host. Well, that's a mouthful. But the bacteria are doing essentially what plants do, right? Plants they harness the energy from the sun, and they use it they harness that energy and they use it to convert carbon dioxide to sugars, right? We're all comfortable with that. These bacteria do the same thing in total darkness, in really cold temperatures by using the chemicals that come out of the cracks in the earth for energy. And what's super cool is that these worms are some of the fastest growing organisms on the planet. It was my PhD research that, through serendipity, which is a big part of science, led us to figure out how to keep these alive in the lab. And basically, I had a water bath fail. And after that, we realized, oh, we've been doing it wrong for about 2 years. And then now we know how to grow them. And I can literally watch them grow. So I'll come in one day, and I'll stain their tubes with a blue dye. And I'll come in the next day, and they'll add ½ inch of tube. Right? And I'll come in a few days later, and they've added 3 or 4 inches of tube. And so and when I measure how much they grow faster than bamboo and kelp. And they're doing this in total darkness. Microbes can also do crazy things like produce their own light. This is a feature called bioluminescence. And these happen to be little bioluminescent algae. I'm guessing this is off the coast of California. But if any of you have had the privilege of seeing this, you know how spectacular it is. But making your own light is a really important way of communicating in the ocean. Microbes are also a part of our own history. And anytime there's a shipwreck, we often find microbes associated with those that tell us something about the shipwreck, believe it or not. In this particular case, anybody recognize this ship? It's the Titanic. It's the bow. And they first discovered these so-called rusticles growing off of it. And this confused people for a while, because when things rust, they normally don't form rusticles. Well, these are iron-oxidizing bacteria that are responsible for that. Right? So Titanic went down, and the microbes that can eat iron suddenly had their equivalent of a whale fall, right? So what are these microbes doing? I've told you there's a lot of them, and they live everywhere. Well, they basically structure our biosphere, and they structure marine ecosystems. Microbes living in the upper part of the ocean where there's sunlight are photosynthetic. And this little microbe called Prochlorococcus is a tiny cyanobacteria that is responsible for about maybe up to half the oxygen that you breathe, right? A more accurate way of saying that is about half the oxygen you breathe comes from the ocean, from microbes in the ocean and algae and the like, and these cyanobacteria are a major part of that, right? And they're so small that for many years people didn't really know how to study them. They kind of knew they were there, but they didn't know how to culture them and so on. And that took a while. Microbes also play a tremendous role in recycling nutrients in the ocean. You heard a bit about that from Joe. And it's particular stuff called marine snow. Now, if you don't know what marine snow is, it's the kind of gooey stuff that rains through water. If you've ever seen a video of a submarine going through the ocean, you see all this white stuff? That's marine snow. It's fish scales, fish poop, more fish poop, boogers. I mean, it's everything under the sun that's produced up in the upper waters that rains down to the bottom that feeds those organisms. And microbes play a role in breaking down nutrients that as it moves through the water column and making them available to those parcels of water. Right? So they play a huge role in maintaining ecosystem health. And in the deep sea, we're beginning to realize that a lot of microbes are producing fresh carbon, kind of the way that the algae do, off of this chemical energy. Now, we've known about that. I told you about that with the tube worms, and we know that they exist, but we're just beginning to realize that this underwater aquifer I mentioned may actually be harboring an awful lot of these. And so there may be fresh food that's being injected out of these holes in the cracks in the earth and feeding deep sea communities to an extent we didn't previously realize. So the punchline is that microbes are really critical to our biosphere's health. Right? They produce half the oxygen you breathe. They fix nitrogen. That's a fancy way of saying they take nitrogen gas, which you or I can't use, and they turn it into something that living organisms can use. And without microbes doing that, this biosphere would come to a halt. You could wipe out every person on the planet, and at the end of the day, the planet will keep running. But if you wipe out every nitrogen-fixing bacteria, this biosphere will stop very quickly. Okay? But their precise role remains to be determined. In other words, we know they're out there and we know they do this, but we don't exactly know who's doing what and each kind of microbe's contribution. But I get asked this question a lot: can microbes save humankind? Now, I would say microbes are one of our best bets for carrying out some sophisticated chemical reactions that we can't carry out. I'll give you an example. Wastewater cleanup. Deer Island depends on microbes. I mean, we don't think about it all the time, but they do. If you killed all the microbes, Deer Island wouldn't work. So wastewater cleanup is inherently dependent on microbes. The pharmaceutical industry uses microbes to produce particular pharmaceutical products. Bioremediation is a hot topic, and we heard some folks today talk about the microbes in the soil supporting ecosystem health. That is absolutely true. If you keep the microbes happy, your ecosystem will be much healthier. But we have to be careful about being too cavalier, because we can't just always turn to a technology, even if it's a biotechnology, to fix our problems. It's too easy to do that and keep up our kind of crummy behaviors. The big issue for us is typically a question of rate. There are plenty of microbes that fix carbon dioxide into sugars, and one could argue that, hey, let's grow all these microbes and have them suck CO2 out of the air. That is a very difficult scenario to imagine, right? And I understand, I am definitely all for the efforts to improve the health of our biosphere to do that, but I just don't see us building a big giant fermenter or monoculture of microorganisms to do that kind of job, because there are some problems. It's about the rate, and as soon as you go to monocultures of any kind, you're susceptible to viruses, right? So microbes are going to can play a huge role in improving the health of our planet, but let's not be too cavalier to think that, that we can enslave them to strictly do our will and have them not be subject to the other forces of nature, like viruses and diseases and everything else. So I have a lot of hope in them, but let's just be mindful of how we use them. So though they're small, though microbes be small, they're mighty, right? And marine microbiology is a wide open frontier. So for you young folks in the audience, this is a really cool place to do work, because there's so much untapped potential, and I mean that in a very literal sense. And their importance can't be overestimated. And it's our ongoing efforts that I hope will help us better understand how to foster the health of microbial communities to make our world continuously habitable. So with that, I'm going to thank you for your time.